Hello and welcome to my poster. My name is Oana Ursu, and I'm excited to tell you about our work on a unified representation of esophageal squamous cell carcinoma cell states and regulation. This project has been spearheaded by Sean Gillespie and my group at Genentech, and it's been a really wonderful collaboration with Nicole Origi, Orit Rosenblatt-Rosen, and Louis Vermeulen. So clinical studies implicate specific biomarkers that are associated with favorable prognosis in cancer. And specifically, understanding these biomarkers requires understanding the underlying cell states that lead to these biomarkers, because that reveals which specific cell functionalities are perturbed in disease and could be targeted for cures. Single-cell transcriptomic studies have measured these heterogeneous cell states within and between patients. However, it's challenging to integrate across different studies because they may be biased by their cohort composition and batch effects. And so in this work, we integrate single-cell transcriptomic data across multiple studies, and we reveal three important results. First, we define the robust axis of variation across patients. We predict putative transcription factor regulators, and then we compare the healthy esophagus with the malignant gene programs that we identified. How do we do this? First, we start with a single-cell transcriptomics collection from two different data sets derived from cancer patients and one derived from healthy esophagus to be used as a reference. We then identify gene programs that are shared between these data sets by first looking for gene programs or co-varying genes within each data set, and then clustering the resulting gene programs across all three data sets to identify these meta programs that are basically consistent across data sets. Then we can compare patient samples from healthy or cancer states. And the way we do that is we take each gene program, we score each cell in our collection, and then we compare the distribution of scores for a given gene program between the healthy esophagus and each disease condition. And finally, to understand what may be driving these gene programs, we assign putative transcription factor regulators to them. The way we do that is first, we start with a database of which transcription factors may regulate which gene, and that is downloaded from Dorothea. And then, given this transcription factor to gene map, we can compute an activity score for each transcription factor in each cell, based on the genes that are predicted to be targeted by that transcription factor. So now for each cell, we have a score for each gene program, as well as a score for the activity of the transcription factor. So then we compute a correlation matrix between the activity of transcription factors and gene programs to reveal transcription factors whose predicted activity, not just expression, but predicted activity, correlates with gene programs. And those are deemed to be putative regulators of these gene programs. It's important to note that this could happen through direct effects, meaning the transcription factor actually regulates the program. It could happen through indirect effects of the transcription factor regulating other transcription factors that regulate the program, or it could be that the transcription factor activity itself and the gene program are regulated by something else. And from this analysis, we cannot distinguish between these three options. So that's why the results that are presented here are preliminary. So let's dive into the results. Here, I'm showing you a unified representation of the healthy and malignant esophagus from single-cell transcriptomics. This is about 50,000 cells spanning healthy tissue and patient samples across 70 different patients. Then, second point, we identify these reproducible gene expression programs across data sets, as I just described. So here I'm showing you a correlation matrix between the scores computed for each gene program across all the cells in our data set. So in each data set, we identify a different gene expression program. Each of that program is then scored across our entire collection. And here is the correlation matrix between all the gene programs. We cluster this matrix and obtain 9 meta programs, 1 through 9. And We study the gene ontology terms that are enriched in the genes of each program. Third, we then take these meta programs that we identified, these 9 programs, and we look at how they vary between healthy and disease conditions. So in this dot plot, each row is a different program and each column is a different condition. They're either cancer patients or they are the normal healthy sample. And so the color here represents the difference in the gene program score between patient and the normal. And the size of the dot represents the significance of the difference. And we can go through each gene program and see that some of them are consistent across all patients, while some programs vary between patients, maybe due to the subtypes or different sort of heterogeneous cell states, these cancers. Finally, we predict transcription factors that may regulate each of the gene programs by looking at transcription factors whose activity is correlated with the gene programs across single cells, where each column is a different gene program and each row is a different transcription factor. So we can then take this heat map and query the specific transcription factors that are associated with the variation in each gene program. So in conclusion, I've told you about our efforts to produce a unified representation of the healthy and malignant esophagus. We identify 9 gene programs that vary across healthy and cancer patient samples. We then identify specific groups of transcription factors that are predicted to regulate each program. And for future work, the field will take these gene programs and study the functional roles of these cell states for tumor growth and progression.